We're going to move into the next session. Just a few comments on the prior session. Incredibly inspiring to see the emerging and current leaders in renal cell carcinoma, and it's changed so much in the past 2 decades. The strength of the clinical, the translational, and the basic science that's in this disease now, I think is really changed a lot for the better. And I think we have a great manifestation of that here at this current session that we're going to be going through. The tumor microenvironment, we've sort of evolved from trying to figure out what the genomics of renal cell carcinoma are, and now we're really moving into more of an integrative biology where we're looking at how the genomics of the renal cell carcinoma interact with the tumor microenvironment and how to really come up with even better treatments. We have an amazing cast of characters here that are going to be giving some presentations. And I also have an amazing co-moderator who requires no introduction, Eric Hakimi, who's going to help with this session. So the way these sessions work and the way the magic of these is that the talks are very short and we have lots of time for questions. So it's an hour, we have about 7 minutes per speaker, and then we have slightly more time, hopefully, for discussion. So we strongly encourage people in the room to ask questions. We're also going to be asking questions. This should be the beginning of a dialogue and hopefully some additional DoD and NIH grants in the future. So let's start off. It really gives me great pleasure to introduce Dr. Eleonora Dondossola, who's going to talk about the bone metastasis immunological niche and unraveling its complexity and its role in therapy response and resistance. Take it away.
Thank you. Thank you for this great opportunity to present some of our insights on bone metastasis and RCC. As you know, bone metastasis is a little complication for RCC patients. Since therapies are working better now, more patients live longer, so they get more chances, unfortunately, to develop bone metastasis. So the rate of bone metastasis is up to 30–40% for metastatic patients. There's a high rate for skeletal-related complications, and these lesions are mostly osteolytic. And this has an impact also on the progression of these patients, of the overall survival that is worse compared to other metastatic sites, except brain and liver. So patients that are symptomatic are often excluded from clinical studies because of their performance status. So they have to undergo radiation or surgery. So they represent an understudied and underrepresented population. Furthermore, accessing samples through biopsies in bone is difficult, and also the processing is complicated. So we don't have much knowledge and much translational research ongoing. So for these reasons, bone metastasis progression and response to therapy remains largely unexplored, and also patient management is suboptimal. In my lab, we focus on the analysis of the interactions that are key between the tumor cells and the bone stroma, and that are important for both tumor progression and also therapeutic resistance. Availability of models for studying bone metastasis, clinically relevant models, is not great. So the first effort that we had was developing clinically relevant models. We study patient-derived materials, and we also have a big preclinical component based on advanced intravital multiphotonic microscopy, tissue engineering in vitro, and also in silico models, mostly agent-based models, and also artificial intelligence. And we apply these models for studying the response to therapy. And this includes targeted radiation, kinase inhibition, so anti-angiogenic TKIs, and also immune therapy. So we received, in collaboration with Dr. Campbell, a DoD Translational Research Partnership Award, and this is a true partnership. So it's a combination between our preclinical skills and the clinical skills of Dr. Campbell. The way we conceive this award is basically we start with a collection of patient-derived material. This is possible through a collaboration with orthopedic surgeons, so Dr. Sacher that is here, and Christopher Alvarez in neurosurgery. So we get access to these bone resections, and our purpose is to combine single-cell RNA sequencing studies, so to get deeper into the complexity and the both cellular and molecular of these samples, and combine these with more topological studies that are based on microscopy. So by integrating these aspects, we would like to prioritize candidate biomarkers, identify pathways of resistance, and also improve treatment efficacy. These samples are very well annotated, so we can match this clinical data with the cellular and molecular data. And we would like to get new insights to be linked to clinical milestones. And of course, this is a back and forward. So we want to link this to prospective clinical trials and also to go back to our preclinical models for studying the complexity of this disease. We started this project in September last year, so we were able to collect 23 samples from bone resections. These are some data related to the patients. Onset of metastasis comes slightly later after the diagnosis of the tumor. These 23 patients, they are all except one clear cell carcinoma, and some of them have either rhabdoid or sarcomatoid differentiation. 1/3 of patients have poor prognosis, and the rest is intermediate, and they present with also metastasis in other sites. So metastases are distributed in both the axial and appendicular skeleton. And what we are doing is also tabulating the all systemic therapies that these patients go through before and after the collection of the bone metastasis. So as you know, the landscape for the treatment of these patients is very complicated. So we would really like to try to understand if there's some correlation. Just to show some more related to the preclinical aspects of our research, one strong model that we have is based on intravital multiphoton microscopy. So what does it mean? It means that we can inspect the disease, in this case renal cancer, and its interactions with the tumor microenvironment, including osteoclasts and blood vessels. So to do so, we developed this model that is based on tissue engineering. So we make a new bone under the skin of the back of the mouse that is suitable for implantation of an imaging window. We directly implant the tumor cells, and we can perform this live imaging in the living mouse. This is how the chamber looks like with this ossicle inside, and these are the tumor cells. So we can capture the progression of the tumor. So here you see a 3D reconstruction that is in white, you see the bone, in green you see the tumor cells, and in purple you see the blood vessels. So we can perform this reconstruction and inspecting the disease in real time. So this is flanked with more conventional approaches that are based on implantation of tumor cells in the tibia. What happens is that when we do this type of implantation, tumor cells can also get to the lungs. So we can inspect together two different metastatic sites. So this is an example for bone and lungs. In green, you see the tumor cells, and in this case, we had some infiltrating T cells and endothelial cells. So in parallel, we are also optimizing some systems for spatial biology of, this is an example for the mouse bone marrow. We will translate these to patient samples. We started with mouse samples because they are more easily available, and we can identify up to 20 markers altogether, and this will allow us to try to dissect the complexity of the immune environment in bone. So thank you very much. I'm open now for the discussion. I have to thank all the people involved, so mostly Matt Campbell and Stefan, that is the postdoc running the experiment, all the people collaborating with us for this DoD project, and the DoD, of course, for supporting us, and all of you for your attention. And I'm open to any questions. Thank you.
All right, we'll open up for some brief questions. Some questions now. Abhishek?
I have to catch this because Tony told us to. So the question is about technically, when you're doing your injections, are you doing intracardiac injections, tail vein injections? You're seeing tropism both in the bone as well as in the lung. Have you tried to compare what if the site of deposition has any impact on the biology of the tumor cell itself?
Yeah, this is a very good question. So we do intratibia injection because it's more reliable. We plan to do also some other type of more systemic injection. So our focus is studying the therapy response. So as you know, if we do intracardiac injection, we don't have a good control of the sites that are interested by the lesion. So what happens is that often soft organs allow the tumor cells to develop faster. So then we don't have time enough to study the biology of the disease in the bone. As an alternative, we have a protocol for intrailiac artery injection that mimics better the systemic distribution of the tumor cells because it's through the circulation, but it's more localized into the limbs of the mouse. So we mostly use the tibia injection. We cannot exclude that, of course, different route of administration of the tumor cell might impact, because if the cells are coming from the systemic circulation, the niche that you recreate might be more physiological, let's say. So for sure, this is an important point that we would like to study. We will also try to compare lesions that we directly implant in the tibia or in the vertebra. Our collaborator, Christopher Alvarez, is working to develop this type of injection that is pretty complex, but that's also in our projects. Yeah.
Take the tumor out from either the bone or the lung after depollution, and it was secondary transplant. Do you see specific tropism towards that, from the tissue from which it was originally recovered?
Yeah, that's a good point. We didn't try yet. So we have a collaboration with Dr. Sacher. They performed a similar experiment after intracardiac. They isolated the cells that colonized the bone, but this was done with human cells. So we have both models ongoing. Since we want also to study the immune system, we are focusing now on murine cells.
I'll have a question, if you don't mind. Wonderful work.
Thank
So you had showed this implantation of human tumors into the mice. Is that what you were?
So we have both models. We have murine cells and we also have
So the
human
murine
cells.
cells, what murine cells are you
We use
using?
Renka. Yeah.
Okay. And my second question is about what your experience has been actually assessing and analyzing human bone metastasis. Historically, they've been tricky to get good cellular composition. Sometimes,
Yeah.
I mean, there's obviously varied. Sometimes you have a huge tumor in the bone, but often they're smaller and more challenging. So how have you overcome that challenge, or what has your experience been there?
So the advantage is in getting surgical resection is that the material is much more. You are right. So the quality of this material is not always good. For single cell RNA sequencing, it's easier because you dissociate, relatively easier because you dissociate the tumor. So you can eliminate the dead tumor cells. The microscopy part is much more challenging because this tissue sometimes is really mostly necrotic. So it's much more complex for us. So this is why we are starting optimizing the system with murine bones and then moving to the patient sample. But yeah, for sure, it's very complex.
Question for you as well. Fantastic work and really, really important. What's your, clearly the immune microenvironment in the bone is different in RCC and it's less responsive in that. What's your sort of working hypothesis on why there are these differences?
So we think that bone resident cells for sure contribute in resistance. For other cancer types, it's known that osteoblasts promote resistance to systemic therapies, also to TKIs. We have seen this in prostate and also in our department, the work of Dr. Sacher, Sualin has shown this. So for sure, these stromal elements are affecting first of all, the resistance of the cancer cells, but I think they also have a big impact on the type of the infiltrate and also on the status of these cells, if they are activated or not.
Interesting. And based on what, I know you've done a lot of work in prostate cancer as well, if you were to sort of think of a potential therapeutic path, if you do validate that hypothesis, how would you approach a new therapeutic paradigm?
So this is what we are trying to do with our models, trying to understand. So I think we have the advantage of studying the phenomenon in the living mouse, so we can monitor the evolution. And for example, also the arrival of the T cells. We see in renal there are many T cells, but probably they are not functional because the tumor keeps on growing. So by being able to study also these aspects that usually are not considered because it's very difficult to study the dynamic evolution, we think we can get insights also on the way the therapies can be combined. I think it's also very important how the administration occurs. So if you give after or later one specific treatment, it can make a big impact, a big difference. So we want to focus also on these specific aspects to understand the schedule of administration and how different combinations can work better.
Fantastic.
Okay, thank you. Wonderful. Next up is a woman who needs no introduction, Dr. Rathmell from Vanderbilt, talking about metabolic demands and determinants in the RCC tumor microenvironment.
All right, thanks very much. I want to thank the organizers for the opportunity to give this talk. This is the first year into also a Translational Research Partnership Award. My partner is Jeff Rathmell, my husband. So to the KCRP, this is our first grant that we wrote together. We survived that, and so it also added a dimension to our marriage. So, so we're studying metabolism. So, and very relevant to the talk that we just heard and the microenvironment and how metabolism influences that. So this audience doesn't need this introduction, but the effect of VHL mutation on cells in the kidney leads to this massive reprogramming, much of which affects metabolism and gives the opportunity for cells to be taking in advanced levels of glucose or glutamine and to really reprogram the way these cells in a dish can utilize various nutrients. But one thing that's really bothered me for a long time, and I'm not going to solve it here, I'll just tell you that, but is why some clear cell renal cell carcinomas, with that knowledge that they all have a VHL mutation, but some can be stone cold negative on a FDG PET scan, like you see there on the left. That's a confirmed clear cell renal cell carcinoma, not taking up any glucose, and some can be blazing hot. There on the right is an example of that. Similar size, very similar histologies on these tumors, and yet there's a big difference. So that got us talking to Jeff Rathmell's lab. He's an tumor immunologist and a T cell. There you go. Oh, that got a lot better, actually. A T cell metabolism person saying, well, you know, your tumor cells are using nope. Oh. Yes. There we go. Okay. But him saying, you know, glucose influences how those cells respond and how they differentiate, making more cytotoxic T lymphocytes if they utilize glucose. And we've got macrophages, which I show there in red, which are these sort of, you know, big burning cells that also live in the tumor microenvironment. And guess what? PET scan isn't a tumor scan. It just measures metabolism in any cells. And so here's a patient who had a COVID vaccine, and you can see a PET response there. So if you're in the tumor microenvironment buffet, per se, who gets to eat the glucose, right? Is it the cancer cell that's been reprogrammed? Is it the T cell that's wanting to differentiate and be more cytotoxic? Or is it the macrophages? And to summarize, I'll get it all up there. This is a paper that we published about 2 years ago. We started taking mouse tumors, Renka and others, and deconstructing them and looking at where metabolites were going and using fluorescent tags as well as radioactive tags, and showing that fatty acids and amino acids were heading to the cancer cells. You can see there graphically shown in the gray bars. But glucose was very consistently going to immune cells, shown there in red, and mostly going to macrophages. So really an interesting separation of cells being hardwired and programmed as to what they were going to choose to eat in that microenvironment. So that led us to go back to the drawing board and say, what is making this difference of what immune cells are present there in the tumor? Why in a tumor are there immune cells and there's not the same repertoire of immune cells out in the normal kidney? And so we took Renka and knocked out VHL because we wanted to ask, is VHL contributing to this overall phenotype? Actually, we have several cell lines. Some we got from Eric Jonasch. We made our own as well. So we have several biological replicates to ask, if you knock out VHL, does that change anything? And then we add it back so that we can really consistently know if the factors that are coming from the cancer cells are actually changing that immune environment. So guess what? If you knock out VHL, you get more tumor-associated macrophages, TAM. So that's what's shown here. You can see if we deconstruct a tumor and then we flow for different subsets, we get actually two different subsets of tumor-associated macrophages that are some that are CD11b high and F480 low. These are what we're calling our TAM1 macrophages. Overall, that if you look in each of these little triplicate bars, it's wild type knockout, higher, rescue, bringing it back down, showing that it's very much a VHL effect. VHL only mutated in the cancer cells. You inject it into the mouse and it's changing the admixture of cells that are present. So there's more macrophages hanging out in these tumors. And they're more glycolytic. I'm skipping a little bit of data that shows that actually the cancer cells themselves were not more glycolytic. The T cells were not more glycolytic. When we look at the spleen, there's nothing happening there that's more glycolytically active. The only cells in that environment that are taking up more glucose than they would have in a VHL wild type tumor were the macrophages. So that's shown here in yellow and also in the magenta color, or more like wine, I guess, is tumor-associated macrophages taking up more glucose in this environment. I don't know if we recruit a more activated type of macrophage or if they come in and become more activated, but they're clearly taking up more glucose. Why? So we're still working to understand that. This is very early days, but when you knock out VHL, a variety of things happen. Lots of things get secreted. We started to look at cytokines. So when you knock out VHL, we want to make sure not just everything changes. This is an example of one of many cytokines that doesn't change, GDF15. So it's wild type, knockout, rescue, no change across the board. Look at somebody familiar, VEGF. We know it should go up when you knock out VHL, it should go down when you put it back. It does that very consistently. And then we found a surprise. And so one cytokine, but there are a couple that we'll be following up, but this one we're interested in for today is CX3CL1. So it's a macrophage cytokine ligand. I'll tell you a little bit more about it in 1 second, that when we knock it out is significantly increased, and when we rescue goes right back down. What is it? So it's a ligand that normally lives on endothelial and epithelial cells, as well as on tumor cells, that is cleaved and secreted to be a very potent inducer of recruiting macrophages into a local environment. It binds to a cognate receptor, the CX3CR1, and brings in those macrophages. And so guess what? When we look at the receptor, we take out these tumors, we do gene expression studies on different subsets. I'll just look at the kind of pink and then purple, how much CX3CR1 is present on the macrophage subsets. It increases in the TAM1 subset when we knock out VHL. It's quite high across the board, actually, in the TAM2s. And so this is our model. I've gone just 30 seconds over. VHL loss increasing the secretion of CX3CL1, being a recruiter for macrophages, bringing them in and also bringing in or creating a more activated subset of macrophages. And that's important. Why? There we go, because what other factors might be influencing macrophage recruitment? This doesn't account for why there are some tumors that are cold and some that are hot, but there are probably many things that can influence how many macrophages are coming in. But can we use this as a biomarker of outcome? Is this adaptable? And can we change it so that we can more accurately harness the immune system when we're trying to target kidney cancer? And so with that, that's the group, more to come, and just a really exciting project diving down under the layers into the microenvironment. Thank you.
Okay, we open up the floor. Dr. Henske.
That's super cool. You said that you don't think this explains the hot versus cold PET scans, but maybe it does. Maybe there's a difference in macrophage biology and might tell us something about the tumors.
Yeah, I do think that it does. I think it's just, it's not all VHL, right? So because both of those tumors were clear cell, both of them would have had VHL mutations. So I think that VHL contributes to a macrophage feature, but my suspicion is that secondary effects, potentially other things that are important in the microenvironment. I'd really love to look at some of this in, bone versus lung. But I think there are a lot of things that can influence how many macrophages are there. But I think that it probably does in part account for that.
Thanks.
Oh, yep.
Abhishek, and then gentleman in the front.
Couple of questions. So one is when you're doing your metabolic studies, I saw that many of the consumption studies were based on per million cells, how much was being taken up.
Yeah.
So can you comment on the composition of the tumor itself in terms of how many immune cells versus tumor cells were there?
Right.
And then unrelated to that, but it's striking that it almost challenges our dogma of the Warburgian effect, right? So when you're looking at a VHL knockout system, have you tested whether putting back the HIF-DPA mutant or putting in a VHL, when you're restoring the VHL activity, if you put in a functionally dead version of VHL, can a scaffolding effect be sufficient to drive the phenotype?
Yeah, those are all really good, good experiments to do. So I will tell you, it does challenge our assumptions in the Warburg, but let me tell you how Warburg did his studies. He took whole tumors and looked at glucose consumption. So he wasn't looking at pure populations of cancer cells. The assumption at the time was that it was really mostly cancer cells. When we look at how many immune cells are present in a tumor in clear cell renal cell carcinoma, there are a lot, and there are enough macrophages to account for some of the difference in PET scans when we go back and try to do a back calculation of how much glucose is showing up in each macrophage and how many macrophages when we try to count in those tumors. So there is enough to make a difference. It is probably a mixture of effects. I think one of the things that's also really interesting, and I didn't have time to show today, when we look in the dish, the cancer cells take up more glucose, like everyone has shown. And when we put them in the mouse and then give FDG to the mouse, the VHL knockouts compared to wild type are not taking up more. So I think there are reprogramming effects that can happen once you're in the context of the microenvironment that we really don't understand, that are going to be super, super fascinating. And part of this grant is then to go on and look at glutamine and look at other nutrients and try to see, how the variation in nutrients influences all this. So there's a million super cool things to do here.
Okay. Go ahead.
Jim next.
Jim.
Yes, go ahead.
For a couple of brief questions. One, maybe I missed it, but do you see the same thing or the opposite thing, when you reintroduce VHL into clear cell renal cell carcinoma cell lines? And if you look at tumors and you look at with VHL mutations, do you see a correlation between the cytokine expression levels and infiltration by myeloid or macrophages?
So yes. So we've only done this in sort of the correlative way, right? Looking at single cell studies and things like that. We do see the same correlation. So to your first question about when we add VHL back, I think I'm not sure I understand that question exactly. What are you?
Well, so the question, if you put VHL back in, do you see a down regulation of the cytokine? So in other words, I think in your system, you were knocking out VHL.
Yeah, we only showed
it
up. So we have not done that yet. This is still pretty fresh data, but that's definitely on the drawing board. And then I think the other thing, we haven't been able to do this in any human systems because we need to have the whole immune competent. We're working with Ari's cells, his LVRCC model, to try to see, if this crosses models, and then we need to look in more human condition.
We'll have Jando and then Rana.
The model you use is subcutaneous or orthotopic?
So we have done this both subcutaneous and orthotopic. There are some minor differences, but the results are consistent, whether we do it subcutaneous or orthotopically. I'm sorry.
The first question was about
yeah.
vascular recruitment for these macrophages.
Yeah. So, I think it's really, kidney cancer's got this really interesting vasculature. And I will say this is totally separate. We're trying to look at human tumors and try to understand where macrophages are fitting in the microenvironment. and they hang out really close to the endothelial cells. So they're very well may be vascular recruitment that is a part of bringing the macrophages in. And I'd love to do some studies with, turning off VEGF or changing the endothelium and see if suddenly, we don't get those same macrophages there.
I have a question related to this
yeah.
aspect. So, did you check if these macrophages have a pro-angiogenic phenotype? There are also markers like Ty2 or CD13.
Yeah, I don't think we know that that well yet. I think macrophage biology, and a lot of people know this more than I do in this room, is really about to revolutionize. The M1, M2 is not nearly granular enough for the various types and behaviors of macrophages. So we're at this point just defining them based on surface markers and giving them labels in that way. So.
And Rana?
If you were to think about the role that macrophages play within the tumor microenvironment, and obviously they have a very diverse spectrum of things that they can do, but they could potentially be more immunosuppressive. Would you, if you were to design a trial utilizing FDG PET as an imaging biomarker, do you think from your data that bright tumors would actually be more responsive to immunotherapy as opposed to cold tumors?
Yeah, I would love to do that study. I would love to do that study.
That's cool.
And I do think that that's how I would like to design it. I think we think about PET scans as like plus minus, but there's a whole gradient and to look at
like
tumor
SUV
responsiveness.
on a continuum.
Yes.
And correlate it with the
see,
response.
yeah.
That'd be really cool.
Yeah.
All right.
Great. Yeah. All right. One last question.
Very quick about the PET positive versus negative tumors. Have you looked at GLUT1 expression in those tumors? And do they,
Yes,
they might,
we did. And we've published on that. And because we had a whole good series of like MR PET. So we, but at the time we really weren't as cognizant of what was going on in the microenvironment. So GLUT1 absolutely correlates. But GLUT1 is present on T cells and macrophages too. So I actually don't know who was making the GLUT1 that we saw there in those more PET avid tumors.
no, but the immunohistochemistry level, when the tumors were removed, would you see GLUT1 expression on the tumor cells in the negative tumors?
So say that. Oh, we have we done that on our current set of tumors where we know VHL status and things, and have better immunohistochemistry and better awareness and multiplex. We have not done that. So,
Okay.
I was referring to a very old study. So.
Excellent. Fantastic presentation. And we're now going to move on to a similar sort of theme here. And Dr. Hartland Jackson is going to talk about identifying novel immune evasion tumor immune networks as targets for clear cell renal cell carcinoma immunotherapy.
Thank you. And thanks everyone for having me here today. So I'm, I guess, the technologist, the PhD scientist here, and my group works a lot on understanding how networks of cells work together and how this changes during cancer development and how this could be kind of a new class of biomarkers and targets. And we do this by specializing in imaging mass cytometry.
So green button to advance.
And the reason I'm here is because of the opportunity to work with this really impressive translational team in Toronto. So through the partnership grant, we've paired up with Tony Finelli's team across the street at Toronto General Hospital and at Princess Margaret. And we're joining them to really add in this new technology and be able to scale some single cell biology to look at large clinical cohorts. So the reason that we're able to expand on is that there's already been a lot of work by the team to look at different single cell technologies such as suspension cytoff or single cell RNA-seq. They've built out kind of what they're considering a gold standard single cell benchmarking study where you're able to look at these same patients with kidney cancer, identify multiple cell populations. And now we're joining the team to look at how these are spatially organized within the tumors. And so from this small number of patients, they're looking at hundreds of thousands of cells. They've really nicely classified the immune environments present in these patients. Now looking at which are actually organized and present in the same groups, the correlation within certain patients. But as you can see here, this is kind of in the 60 marker scale, which is really nice, but you don't know just because they're correlated, are these actually interacting with each other within the same tumor? And so our goal is to use imaging mass cytometry where we can look at over 40 different markers at the same time in a single FFP tissue section to identify how these groups of cells are organized, how they are working together in renal cancers. So imaging mass cytometry, IMC, it uses very similar staining protocols to what exists for immunohistochemistry. The difference is the antibodies we are using have each been labeled with a specific metal isotope of a certain mass. And then kind of scary if you're used to doing immunofluorescence is you actually dry out the sample completely, and then you introduce it in the laser ablation system where a laser then rasterizes your tissue. So there's 1 micron spots of tissue that are kind of aerosolized and then atomized and ionized into the mass spec. You then get every laser shot becomes 1 mass spec readout. And then, so in that 1 laser shot, we can measure over 40 different metal isotopes representing each of the antibodies. And then that laser shot becomes 1 pixel in the images that we can recreate. So the reason that we're able to use this technology to look at a lot more patients is for a couple of reasons. One, this is imaging on FFPE sections. So there's no tissue dissociation, no cell loss that's taking place. What the pathology team gives me on a slide, we can actually measure and we get 100% of those cells. There's now a lot of studies coming out showing that you do get enrichments of different cell types from dissociation, and we're able to kind of bypass that, go after these fixed tissues. That means you can give us really small or rare samples and we are going to capture the data from it. No problem. If we do a whole slide, we're looking at then millions of cells per sample, not just a few. And we can go back in time. We can go back to those biobanks that all the hospitals have. We can select certain patients and then profile the immune cells in those tumors, even from samples that are upwards of 20 years old. And a unique feature of IMC and these metal stains is that they're actually stable. So you can put the antibodies onto your slide, dry them out, and we can have treated them kind of badly in the research environment where they sit there in a drawer for 6 months to a year and the signal's still great. The reason is there's no enzyme in there. There's no fluorophore that's going to break down. As long as the metal is sitting there in the tissue, we can still get a really good stain. So this allows us to do large batches and standardize these stains across a lot of samples. So the reason we're also able to do this is because the lab has 2 dedicated imaging CyTOF systems. So huge capacity for about 15 people. I'm seeing I got to speed up. This is looking at the laser ablation on the actual tumor. So you're seeing it kind of carves off 1 micron along the bottom, and then you digitally reconstruct that image. And so what this looks like in RCC is you'll see this is going to flash by. This is all 1 measurement from 1 kidney cancer. So if it will play, oh, we skipped it. Let me try again. Nope. Anyways, it flashes 30 beautiful markers at you, but you can also look at these as in combinations. So all of this is present in actually 1 sample. So even if I'm showing 2 images here, this is 12 of the 30 plus markers. We can see all the cell types at the same time. In our preliminary studies, we're also looking at 5, 6 TAM phenotypes and how these are organized in these tumors. In the past, what we've done with this is to look at large clinical cohorts and tissue microarrays, count all the cell types that are present. But beyond just saying these cell types are in a tumor, we see where they are, where they're organized in tumors. And so we could see that even the same cell types in breast cancer seem to be organized in different ways. And so to quantify this, you can actually identify which cells are interacting with each other and then use analysis to identify communities of cells, whether this is tumor communities or tumor microenvironment immune communities. And we could see that in breast cancer, these features, the organization of these cells actually provided clinical outcome related features more than just the single cell populations. So the goal here is to work with the clinical team at Toronto General. We're looking at building out TMA cohorts right now that we're able to do 2 different cohorts, discovery cohort of 350 patients, 350 validation. This means we're able to produce about thousands of images with 45 plex measurements, count the immune cells in these, look how they're organized, and identify are there networks of immune cells, not just that the cell is there, that's really related to outcome. And is this what we're targeting? Is it a group of cells that's actually responding to immunotherapy? So further, what we're doing going forward is we want to see, can we now have functional models to do this as well? We need to go to humanized mouse models and other mouse models. And the only reason able to do that is the lab has also created mouse immune profiling antibody panels to do 43 markers in mouse tissues all at the same time. So the idea would be, can we identify models that also have the same organization of immune cells and target these? So all this is really due to us being able to pair with clinical teams that have spent decades putting these samples together, and also being part of kind of an extended, really collaborative community in Toronto. This is across 5 or 6 different institutes, all within one block, people all working together with different expertise in mouse models, clinical samples, technology, everything that we can do. So I look forward to your questions, and thanks for having me today.
Beautiful work. Questions from the audience? I'll ask a quick one. What is the cost of your assay comparatively to other?
So it depends. We have the benefits of kind of some scale. And so if you're collaborating with me, you're not taking on any of the instrument time or other things like that. Once you have the machines themselves can be expensive, but on the range of high-end microscopes, for example. But they can run 24/7, which is nice. That helps with time. And then the consumables are just gas, et cetera. So if I'm going to one of these, I can do a sample for $500 probably, would be generally pretty good, which allows us to do larger scale, which is good. But it's still in the very research phase. We're still developing all the reagents ourselves as well. So we do take commercially available antibodies and label them with the isotopes themselves. And so panel development has its costs as well. Purified antibodies plus isotopes.
Right, Lisa.
Ask about the famous tumor heterogeneity question, and if you've applied that to the breast cancer models, and how many different specimens from an individual tumor do you think you need to sample to get an accurate idea of what's going on?
So this is a challenging question, also a fun one, also where we kind of live and think all the time. From our experience, it really changes between different cancers and between different patients. And so in some cancers that we see, the tumor heterogeneity is the tumor itself can be kind of massively spatially heterogeneous. We've seen tumors where the immune system, there are different immune environments in different locations, but also those where the tumor cells are very heterogeneous, but the immune content or the immune environments is actually homogeneous across the tumor. So I kind of think this is related to the stage of tumor development or the type of tumor development. Are there different clones that are being selected for and in an ongoing battle with the immune system, or if they kind of bypass this early and you're seeing a lesion that has already suppressed the immune system and gone past it? And so there are definitely different types. There's immune heterogeneity, there's tumor heterogeneity, and relationships in between. But there are homogeneous tumors. And I think that's actually really helpful for the idea of being able to treat them, because that means that it's, even if you sample a biopsy and make a decision based on that, that it does apply to everywhere. In some cases, no.
Experience and based on our morphologic assessment as pathologists, we often see intense morphologic heterogeneity in kidney cancer. I think everyone in this, I think that's what you're alluding to as well. And what we saw in our CP-Tech experience was the fact that renal carcinomas, which are inherently heterogeneous, or they have more than 4 different types of morphologic patterns under the microscope, tend to do worse than less than 4 morphologic patterns. Currently, none of the cancer templates as reported by GU pathologists or as needed by medical oncologists or urologic oncologists take into account this morphologic variation. I do think we have done one follow-up study where we had similar experience that high morphologic variation is associated with poor outcome. And then in the CP-Tech experience, also the whole genome instability, BAP1 mutations, high feature counts, epigenetic modifications were all associated with this. So I'm just wondering, in terms of the massive amount of data which can be produced by a single section over here, and I think you're planning to do about 300 tumors. What is the plan, or how do you reconcile all of this immense information which is produced by this massive data set with the genomic parameters, which I assume are available for this cohort?
Yeah, so in the past, what we've done is you are taking a different piece of the tumor for the two, but you can then correlate across features. And we have seen really nice relationships in breast cancer between the genomic profile and the types of cells that are present there. And so it's correlative, but this is the place to start. You're then identifying which genomic relationships are present to a certain immune environment, for example. This is why we're building out the immune models. We then have the mouse models. We then have systems where we can induce and functionally test, is this a genomic driver of certain immune phenotypes? And is there a relationship between these two things in terms of resistance or what type of environment is produced? And can we test these? Can we intervene along those lines? In terms of the spatial heterogeneity, I think we also do a lot of spatial transcriptomics and other studies where we're looking at how this changes. And it is a concern. You're not really fighting one tumor. Maybe you're going to diagnose that patient as this type, but in a lot of the cancers we see, even if there's a dominant phenotype, there are always the heterogeneity that's there at the time of treatment. It is always resistance mechanisms are already present. There's other things. You're actually fighting multiple types of tumors at once. And if you can surgically resect it, sure, you simplify the problem. But I think in general, this is where iterative therapies or other things are absolutely necessary, and why we can't win with a single shot, because it's not one thing that we're fighting. And so how to quantify this and map the space and what the relationships are, so you know what the resistance possibilities are, will help us make clinical decisions going forward in terms of what's step one, what's step two, or what's the best probability at step one.
One quick question. Are you doing metastases as well?
Not in this case, but I'm obviously open to lots of things. So maybe we'll have a chat at one.
Sounds good.
Okay, great. And our last talk is with Dr. Dere from Baylor on targeting combined VHL and 3P chromatin remodeler deficit in RCC.
Thank you. I'd like to begin by thanking everyone for being here and for the invitation to speak. And I think I'm going to switch from more a clinical perspective to more a basic science perspective, telling you a little bit about what we have going on, where we're looking at the combined loss of VHL and 3P in RCC. Okay, so as we all know, 3P loss is accompanied by the biallelic inactivation of VHL, and these are sort of the early events in RCC progression. And loss of 3P is accompanied by a loss of several chromatin modifying enzymes. These include SETD2, PBRM1, as well as BAP1. And these enzymes work in the nucleus, modifying what we call the histone code. So these are post-translational modifications that are added to histone tails. And you have a trifecta of enzymes that carry out these reactions. You have the writers, readers, and erasers. So these enzymes, as the name suggests, are either writing the post-translation modification, reading it, or then erasing it. Now, a similar code also exists on the cytoskeleton. It's called the tubulin code, where you have microtubules that are marked. And here, SETD2 has been shown by Cheryl Walker's lab, also at Baylor College of Medicine, to be a writer writing a trimethyl group on microtubules. And this trimethylation is in fact read by PBRM1, which is also commonly lost in RCC. And so, although SETD2, PBRM1, and BAP1 have been studied quite extensively independently for their chromatin function, really, they do not share a convergent etiology on chromatin. What we do now know from work of several people is that all of these enzymes do in fact localize at the mitotic spindle. So you have VHL, which stabilizes microtubules. You have SETD2, like I mentioned, it's methylating microtubules. PBRM1 reading the microtubules, and we have BAP1, which is a D-ubiquitinase. It removes this ubiquitin moiety, which is laid down by BRCA1 on gamma tubulin, which is also found at centrosomes. And so the question we wanted to ask or address in my lab was, is there a functional convergence of these enzymes at the mitotic spindle? Why are they there? And can we reveal and exploit any vulnerabilities in this system? And so the first piece in that puzzle was to link VHL and SETD2 at the mitotic spindle. And this was through an enzyme called Aurora kinase A. Aurora kinase, we had shown previously, was marked by VHL for degradation. And so using some in vitro kinase assays, we were able to show that SETD2 was in fact a target for Aurora kinase. We did some mass spec studies to identify the site of phosphorylation, and this was the serine 2080. We've generated an antibody, and then when we stain cells with this antibody, we see phosphorylation at the spindle poles and at the mid-body, which is the picture on the right. And this is where we would expect to see SETD2, because this is where we see methylation, as I mentioned, which occurs at the mitotic poles or along the mid-body. We then went on to ask a question of whether Aurora was phosphorylating SETD2 at the spindles themselves. And to do this, we took human kidney cells and used an Aurora inhibitor. The inhibitor that we used here was called alisertib. And as a readout for treatment, we looked at phospho-Aurora. Aurora is a kinase that autophosphorylates itself. And so you would expect decreased phosphorylation of Aurora upon treatment with the inhibitor. And shown here in green is the phospho-Aurora over dose that seems to go down. And phospho-SETD2 sort of correlates, and you see this decrease in phospho-SETD2. We next wanted to see whether this decrease in phosphorylation, affected or impacted SETD2 activity on microtubules. And here, this is kind of hard to see, but if you look at the vehicle, what we've done is we have stained for methylated tubulin in green, and you should focus on the mitotic poles, and our antibody cross-reacts with the chromatin as well. And that's the reason why you see sort of that band in the middle. But we're really focusing on the poles. And what we see is that with increasing doses of Aurora inhibition, we see a decrease in methylation, suggesting that this phosphorylation was in fact driving SETD2 activity. And this was all fantastic. But the real question that we wanted to get to was, what is this sort of combined impact of losing these chromatin modifiers at the spindle? And so to do this, we partnered with Dr. Ian Frew, who's at the University of Freiburg, and he has developed a RP tech model where he's able to monoallelically chop off a 5.5 MB piece of genomic DNA. And this is then followed by a biallelic inactivation of VHL. And so this gave us sort of the perfect system to ask the questions that we wanted to ask using the system. And to begin with, we asked very simple sort of questions. What happens when you lose VHL in addition to 3P loss? And here we started with just looking for micronuclear formation, which is a reporter for a genomic instability. And what I have on the left here is a cell that's proficient for both the 3P as well as VHL. In the middle, I have a 3P monoallelic loss with VHL wild type, and you don't see much micronuclear formation here. But what's striking is that when you lose both 3P as well as VHL, you see this huge increase in micronuclear formation. What we've also done is looked at the methylation of microtubules that I just mentioned in both of these cell types, where we have 3P loss with VHL proficient cells or 3P loss in VHL deficient cells. And what you see is this huge increase in methylation of the spindle in cells that have lost VHL. And I'll tell you why this is important in just a second. What I've shown on the right here is a quantification showing you an increase in the ROI area as well as the sum intensity of that methylation. So these are sort of initial studies. We've just started a systematic evaluation of mitotic defects, the spindle architecture, segregation defects, centrosome numbers, as well as genomic instability and micronuclei. So to summarize, what I want you to take away from this is, we all know that RCC is not really a highly proliferative disorder. So why study mitosis? I think this is really important because what is really striking is that it's the quality of mitosis that's important and not so much the quantity. So for example, when you lose SETD2 and you don't have methylation of your spindles, technically these are supposed to die. This signal is very apoptotic. And the fact that you have cells that survive this and move on suggests that there are compensatory mechanisms in place, which are sort of enabling these cells to get through that first initial push of mitosis to then develop later mutations. And so our focus is to try and understand what these effectors are during mitosis, so that we can then hopefully exploit some of these vulnerabilities as we identify them. Last but not the least, I'd like to thank all the people in my lab that have worked on it, and my mentors and colleagues now, Kim and Ian, who have been instrumental in some of this work, and the DoD for 2 grants that support this work. And thank you all.
Go ahead.
Thank you. Did you investigate if this is also phenotype in invasion or migration of these cells? We haven't done that. So we've just gotten the cells and, we're just progressing with all of this. But yes, that's one of the things that we will be looking, because what I did not mention is that SETD2 is also methylating the actin cytoskeleton. And so that's where the invasion and migration, I think, will be very important. And another question is about the rigidity of these cells. So if you expect them to be more or less rigid because of these changes? So I think they're going to be more flexible. They're not going to be as rigid. Yeah. But we have not tested that at this point. Thank you.
So sort of in the clinical realm, or the chromosomal copy number changes associated with these secondary mutations, SETD2, BAP1, PBRM1, et cetera, are there some unique sort of chromosomal copy number patterns that you see when you have those?
So we are in the process right now, Eric, of karyotyping these cells to see what exactly we would expect. But in terms of other mutations and chromosomal rearrangements, actually some of those are also sort of feeding into the mitotic spindle and sort of this nexus that we're finding at the mitotic spindle. So hopefully, as we move forward, we'll have more to sort of tell you about.
Just a quick question. You had showed SETD2 and the micronuclei that you had mentioned. So I'm just thinking from an immune and therapeutic standpoint, the association of micronuclei with CGAS STING pathway and potential sensing there and for innate response. Do you think there's a potential link there, and is there opportunity to explore that in your mind?
I would love to say there is. We don't have any evidence to say if there is or there is not. The sad part about these particular cells is that they're not tumorigenic in mice. And so there are other opportunities to collaborate with a few other people who might have similar models.
Abhishek.
I have a technical question about how are you inactivating VHL in your RPTECs? Is it just one single guide, or are you cutting at both ends of the gene?
Wow, that is an Ian question, but I believe the answer is we're cutting in both ends.
I think that would be really important to know, because the rescue that you're seeing, if it is simply because of an in-frame fix in a single cut site, you really just would have restored VHL activity back in what you're thinking is a VHL deficient cell, right? So it would be important to address that. The other thing would be if the cut site is at the 5' end of the gene itself, we know that you could still make the short form in frame, and which could still be functionally active.
Right. So I think protein wise we've checked, and we do not see either the long form or the short form. So that we've verified already.
Just a quick comment about your comment. We have a paper that's in press actually showing that SETD2 mutations do increase STING signaling, and we did not actually look at micronuclei. So it'd be actually kind of an interesting follow-up experiment.
So I think it's appreciated that 3D LOH at the arm level is really intense. But there's quite a bit of evidence actually that SETD2 is haploinsufficient.
Yep.
Other contexts. So it seems like your model is a really interesting opportunity to test how dosage of these core suppressors are acting classically. They actually be acting haploinsufficient manner, even when they're not neutral.
Yep.
Because even when one thing that I thought about Genomics for a while is that actually selection for 3D LOH is really, really strong. So you're always trying to use one copy of these genes regardless of what regardless of what you have the other patient.
Right.
Have you investigated, for example, the model and not have other cognitive.
So we haven't gotten that far, but yes, that's actually one of the things. So we have 7 different cell lines or models that we're going to test, each with the monoallelic and wild type copy and monoallelic with the second copy gone. So yes, that's all sort of, we hope to establish what we would call like the cytoskeletal signature, knowing what happens to sort of some of these phenotypes when we have exactly those combinations.
Great. Fantastic.
Oh, yeah.
I don't need that, no. But thank you so much, speakers, and all of the amazing questions. This is, I think, really an example of how far we've come in understanding the microenvironment. Looking forward to seeing next year's talk, which is probably going to be even better than this year's.